Okay, question number 145. Match the following column and select the correct options. And here column 1, first one is Clostridium butylicum, second one is Trichoderma polysporum, C is Monascus purpureus, and D is Aspergillus niger. In column 2, Roman 1, cyclosporin A, Roman 2, butyric acid, Roman 3, citric acid, Roman 4, blood cholesterol lowering agent. And if you observe, first one is Clostridium butylicum, the name itself indicating that the species name, it produces butyric acid. Trichoderma polysporum, it produces a bioactive molecule, that is cyclosporin A, that is used as a immunosuppressive drug in case of patients who underwent organ transplantation to suppress the immune system. And next one is Monascus purpureus. It produces a bioactive molecule, that is statin. Statin is a bioactive molecule which is useful for reducing blood cholesterol. That means it is not directly reducing the blood cholesterol. It is involving as a competitive inhibitor to inhibit the enzyme which is responsible for synthesis of blood cholesterol. And D, that is Aspergillus niger. This Aspergillus niger produces citric acid. The right option for the question number 145, the correct combination is third option. And next one is question number 146. In water hyacinth and water lily, pollination takes place by. So here the given options, first one is insects and water, second option is insects or wind, third option is water currents only, and fourth option is wind and water. Here very important point, what we are discussing, water hyacinth means it is Eichhornia, most invasive weed of India and throughout the world, and water lily means it is nymphia. Both of them are aquatic plants. So it is Eichhornia is a free floating hydrophyte and water lily is a rooted hydrophyte with floating leaves. But very important, even though they are in water habit, they are not pollinated by water. Very less number, at least only 30 genera. Most of them are monocots will undergo pollination by water, but not all the aquatic plants. Most of the aquatic plants undergoes pollination by either insects or wind. The right option for question number 146 is that is second option. And question number 148, and if you observe question number 148, identify the wrong statement with regard to restriction enzymes. First option, sticky ends can be joined by using DNA ligase. Yes, DNA ligase is useful for joining of DNA fragments, no doubt at all. And second option, each restriction enzyme functions by inspecting the length of the DNA sequence. Yes, during action of restriction enzymes on DNA molecule, three sequence of events are present. One is inspecting the length of the DNA, second one is binding the DNA to the palindromic sequence, and third one is cutting. Inspection, binding, cutting. These are the three important events involved in action of restriction enzyme action. And third one is they cut the DNA strands of DNA at palindromic sites. Yes, exactly correct. Because those are endonucleases within the DNA, they will cut the DNA molecule, particularly in specific palindromic sequence. Fourth option, they are useful in genetic engineering. Yes, after discovery of restriction endonuclease only, it became possible to discover restriction reconstruction of rDNA. That means here four statements are looking correct, but very important point you have to observe the first statement, that is regarding ligases. But what is about the question? Question is regarding restriction enzymes but not ligases. That means the statement which is not related to restriction enzymes is first option. And next one is question number 149. In gel electrophoresis, separated DNA fragments can be visualized with the help of. So how That means what is the staining process of DNA in gel electrophoresis? The DNA is stained with ethidium bromide, and even though after staining with ethidium bromide, they are not directly visible to the naked eye. They must be exposed to the UV rays, then only they appears in the form of bright orange color. So the visualization of DNA fragments in gel electrophoresis is possible only after staining with ethidium bromide and exposing to UV radiation. That means the right option for question number 149 is third option. And question number 152, if the distance between two consecutive base pairs is 0.34 nanometers, 0.34 nanometers, or in terms of angstrom units, 3.4 angstrom units, 3.4 angstrom units, and the total number of base pairs of DNA double helix in a typical mammalian cell is 6.6 into 10 power 9 bases. That means if you calculate, then what they are asking, then the length of the DNA is 2.2 meters approximately. That means in a typical eukaryotic cell or human body cell, the length of the DNA is about 2.2 meters. The right option for given question number 152 is fourth option. Next question is 153. And experimental verification of chromosomal theory of inheritance was done by first option, that is Morgan, second option Mendel, third option Sutton, and fourth option is Boveri. See here, very important point we have discussed. Chromosomal theory of inheritance is proposed by Sutton and Boveri. That means they are stated that the behavior of chromosomes during cell division is parallel to the behavior of Mendelian factors. Based on that, they are studied their proposed chromosomal theory of inheritance. But experimental verification of this chromosomal theory of inheritance is conducted by Morgan, by conducting experiments on Drosophila melanogaster, that is fruit fly. So the right option for given question number 153 is first option. Next one is question number 154. Choose the correct pair from the following. Exonucleases makes cut of a specific position within the DNA. So exonucleases are isolating the DNA nucleotides from the ends, not within the DNA. That is not right combination. Ligases join the two DNA molecules. So what is ligases? Ligases are also called as molecular stitchers or molecular glues. These are useful for joining of DNA fragments. That means right combination. That is question number 154. The right option for that one is second option. And next one is question number 157. And which of the following statement is not correct? First option, genetically engineered insulin is produced in E. coli. Yes, that is the first product produced which is useful for human welfare, or first therapeutic released for human welfare is genetically engineered E. coli, which produces humulin. Insulin produced by genetically engineered E. coli bacteria, we can call it as humulin. And second option, in man, insulin is synthesized as a proinsulin. Yes, we know that the proinsulin consists of three chains, that is A chain, B, C chain, and B chain. This A, C, B chain, three chain polypeptide chain itself is called as proinsulin. And third statement, if you observe, the proinsulin has an extra peptide called C peptide. Yes, this is extra peptide. And fourth one is the functional insulin has A and B chains linked together by hydrogen bonds. See here how this functional insulin formation occurs. If you observe, this is A chain and this is B chain, and here this is C chain. After folding, in between A chain and B chain, there is a formation of disulfide bridges or disulfide bonds. That means the bonds which stabilizes the active insulin is after removal of C chain, it is disulfide bridges or disulfide bonds, but not hydrogen bonds. That means the incorrect statement in question number 157 is fourth option. And next one is question number 163. If you observe in case of 163, which of the following statement is correct? First option, adenine does not pairs with thymine. That is wrong. Adenine always pairs with thymine with the 2 hydrogen bonds. Adenine pairs with thymine with 2 hydrogen bonds. That means second option is correct. If you observe third option, adenine pairs with thymine with through 1 hydrogen bond. Fourth option, adenine pairs with thymine through 3 hydrogen bonds. That means the right option for this one is adenine pairs with thymine with 2 hydrogen bonds. Guanine pairs with cytosine with 3 hydrogen bonds. According to Chargaff's chemical analysis or base pair rule. So the right option for question number 163 is second option. And question number 164. And if you observe, which is the important site of formation of glycoproteins and glycolipids in eukaryotic cell? And if you observe in case of first option, that is polysomes. What is polysome? That means mRNA with many ribosomes, we can call it as polysome or polyribosome. So that is not responsible for glycoprotein and glycolipid synthesis. Endoplasmic reticulum. Endoplasmic reticulum is the site of synthesis of proteins and lipids, not glycoproteins and glycolipids. Rough endoplasmic reticulum is useful for synthesis of proteins because that is studded with ribosomes. Smooth endoplasmic reticulum is useful for synthesis of lipids. But after synthesis of these proteins and lipids, they are transferred to the Golgi complex. Golgi complex is a packing cell organelle where these proteins and lipids are undergoing modification. They undergo glycosylation and glycosidation. That means there is a formation of glycolipids and glycoproteins. So the cell organelle responsible for glycolipid and glycoprotein synthesis is Golgi bodies. That is question number 164. The right option is fourth option. Next one is we have discussed about question number 165. Which one of the following is the most abundant protein in the animals? It may be both zoology and botany, but no matter it is, we can discuss in case of biomolecules. The most abundant animal protein is collagen, which is an intracellular ground substance. Most abundant plant protein, not only that, most abundant protein in the entire biosphere is rubisco, but animal protein is collagen. The right option for question number 165 is third option. Question number 166. The plant part, which is consists of 2 generations, one within the other. So students, this is very important. We have discussed here. If you observe, the first option is pollen grain inside the anther. Let us see here. This is TS of anther. If you observe a transverse section of anther, this is TS of anther, and it is having 4 pollen chambers. At maturity they fuses. Inside these pollen chambers they contains pollen grains, and pollen grains belongs to male gametophytic generation. Male gametophytic. And what is about anther? Anther belongs to sporophytic generation. That means one generation material within the another generation plant material, gametophyte within the sporophyte. That means statement A is correct. Second one is germinated pollen grain with 2 male gametes. So this is a pollen grain which is germinated and it contains 2 male gametes. These male gametes are belongs to gametophytic generation, and pollen grain is also belongs to, this pollen is also belongs to gametophytic generation. And that means this is not correct. And seed inside the fruit. And if you observe seed inside the fruit, so this is, you consider a mango fruit I would like to represent here. And this is a seed, and you consider this is a mango fruit. This is sporophytic, and seed is also Sporophytic. That means both of them are belongs to sporophytic generation. And fourth option, embryo sac inside the ovule. Here I would like to represent a ovule. Inside the ovule, it contains a female gametophyte or embryo sac, and this is female gametophyte. Ovule belongs to sporophytic generation, and embryo sac belongs to gametophytic generation. That means statement A and statement D are correct. That means the right option for given question number 166 is 1. Next one is question number 168. What is 168? The transverse section of a plant shows following anatomical features. A. Large number of scattered vascular bundles surrounded by bundle sheath. That means atactostele. Atactostele is the characteristic feature of monocot stem. Many vascular bundles are present. They are scatterly distributed surrounding the vascular bundle. A scleran camatous bundle sheath is present. That is why those vascular bundles are called as fibrovascular bundles. But students, very important point here you have to remember. In case of dicot stem, limited number of vascular bundles, they are arranged in the form of a circular ring. That we can call it as eustele. And second one is parenchymatous phloem, large conspicuous parenchymatous ground tissue. Yes, in case of monocot stem, there is no separation of cortex and endodermis because cortex and stele, because endodermis and pericycle are absent. So the entire interior of the monocot stem is occupied by parenchymatous ground tissue. That statement is also correct. And we can discuss vascular bundles are conjoint and closed. In case of monocot stem, vascular bundles are conjoint, collateral, closed vascular bundles are present. In case of dicot stem, vascular bundles are conjoint, collateral, open. What is the meaning of open and what is the meaning of closed? Open means in between the xylem and phloem chemim is present. Closed means in between the xylem and phloem chemim is absent. That statement is also belongs to monocot stem. And D option, that is phloem parenchyma is absent. Yes, in case of monocot stem, phloem parenchyma is absent. That means all the above mentioned statements related with the monocotyledonous stem. That means the right option for question number 168 is second option. And question number 170, the sequence that controls the copy number of the linked DNA in a vector is termed as. Here only I would like to represent the structure of PBR 322, that is plasmid Bolivar Rodriguez 322. In this case, here this is ORI, and this is ROP, and this is ORI, and this is ampicillin resistant gene, and here it is tetracycline resistant gene, This is ampicillin resistant gene. These two are selectable markers, and this is ORI. This ORI site, origin of replication, is useful for initiating the replication process as well as controlling the copy number. That means during gene cloning, the production of multiple copies of desired gene is called as gene cloning. That cloning process is controlled by this ORI sequence. And what are the given options if you're observed? First one is recognition site, second one is selectable marker, third one is ORI site. So ORI is responsible for controlling the copy number. And next one is question number 172. See, here we can discuss 172, identify the basic amino acids from the following, and very straight question from biomolecules. This is second question from biomolecules if you observed. And first one is valine, that is a neutral one, and tyrosine, and glutamic acid, that is acidic amino acid, and lysine is the basic amino acid. That means the right option for question number 172 is fourth option. Next one is question number 173. Match the following with respect to the meiosis. So they are given various stages of meiosis. In the column 1 they are given different stages, and in column 2 they are given the significant events which occurs during meiosis, particularly prophase 1 of meiosis they are asking. And in column 1, A, that is zygote, B, pagotine, C, diplotin, and D, diakinesis. Students, if you observed from cell division only they are given, this is fourth question. So here we can discuss zygote, pairing of homologous chromosomes occurs. That process is called as synapsis. That means the right combination for A is Roman 4. And pagotine, that during pagotine there is a crossing over. That means exchange of genetic material between non-sister chromatids occurs. That process we can call it as crossing over. That crossing over is the most significant in all sexually reproducing organisms. That process occurs during pagotine substage of prophase 1 of meiosis. And diplotin, during diplotin, chiasmata are clearly visible because due to strong repulsive forces, the homologous chromosomes are trying to move away from with each other, but they are binded at the place of physical contact of the two non-sister chromatids. The place of contact of non-sister chromatids which appears in the form of X-shaped structures are called as chiasmata. Chiasmata are formed during pagotine. Chiasmata are clearly visible during diplotin stage. And diakinesis is the stage where all these chiasmata are moving to the telomeric region. That movement of chiasmata to the telomeric region occurs like a zipper-like manner. This zipper-like movement of chiasmata to the telomeric region is called as terminalization. That is why the right combination for question number 173 is third option. And next one is question number 178. That is BT cotton varieties that was developed by the introduction of toxic genes of Bacillus thuringiensis is resistant to. That means the question is regarding BT cotton is resistant to which organisms. We know that from Bacillus thuringiensis, crygenes are isolated. Crygenes means crystalline protein producing genes. These crystalline proteins are toxic to the insects and pests. Insect pests, that is why BT cotton is a insect pest resistant variety. How? Because these crygenes are producing crystalline proteins. Those are protoxins. Protoxins are not active. They are not causing any harm. When they enters into the insect gut region, the alkaline pH of insect gut favors to convert protoxins into toxins, active toxins. They are infecting the epithelial cells of the gut of the insect, and swelling of epithelial cells, formation of holes. Ultimately, eventually it leads to the death of the insect. By that the plants are protected from the insects. That itself is called as bioprotection. And it is a genetically modified plant produced by using crygenes isolated from Bacillus thuringiensis. That means this BT cotton variety is resistant to insects and pests. The right option for question number 178 is second option. And question number 180, that is the last question. And identify the incorrect statements. First option is due to deposition of tannins, resins, oils, etc. Heartwood is dark in color. Yes, heartwood is dark in color because this is the central part of the tree trunk. If you observe, the central part of the tree trunk contains heartwood. It is inactive wood, not useful for conduction of water and minerals because the lumen of the xylem vessels are formed by the tylosis. Tylosis are formed by xylem parenchyma into the lumen of xylem vessels. By that the xylem vessels lumens are blocked by tylosis. That is why they are not able to conduct, and due to the deposition of all these ergastic substances, it becomes dark in color. And second option, heartwood does not conduct water, gives mechanical support. Yes, it is dark in color, not useful for conduction but useful for mechanical support. It is hard and durable in nature and showing resistance against to the termites and pests. And third option, sapwood is involved in conduction of water and minerals from the root to the leaf. Yes, the only active wood is that is sapwood. Sapwood is present in the peripheral. It is light in color. It is useful for conduction of water and minerals. And the central wood is heartwood. That heartwood is dark and non-functional. And fourth one is sapwood is the innermost secondary xylem and is lighter in color. So the second part is correct. So that is sapwood is light in color, no doubt, but it is not innermost. Innermost wood is heartwood, but not sapwood. That means the fourth statement is incorrect. That means the correct option for question number 180 is fourth option. Thank you, students, and I hope all of you are able to follow this one and this analysis, and wish you all the best, all the students. I wish all the students to get the desired rank and expected college, medical seat in expected college. Thank you.